Hi, Assalamualaikum, and today we will learn about Biology, Chapter 6, which is 6.4, Food Digestion. The food we consume are complex organic molecules which are too large to pass through plasma membranes. In order for food substances to be used by the human body, they have to be converted into a form that can be readily absorbed by the body cells. So what is digestion? Digestion is the process that breaks down complex food substances into simpler, soluble molecules that are small enough for the body to absorb. Food digestion takes place in the alimentary canal. Alimentary canal is a long and muscular tract which extends from the mouth to the anus. So this is the structure of the alimentary canal. It consists of mouth, pharynx, esophagus, stomach, small intestine, large intestine, and anus. The small intestine constitutes 2/3 of the length of the alimentary canal. So now I will tell you the details about small intestine and large intestine. Small intestine, it consists of duodenum, jejunum, and ileum. Meanwhile, large intestine, it consists of colon, which are sigmoid colon, descending colon, transverse colon, and ascending colon, cecum, appendix, and rectum. This is the most important part. Digestion breaks down carbohydrates into glucose molecules, proteins into amino acids, lipids into glycerol and fatty acid. These essential substances are required by the body cells to carry out metabolic processes. For example, glucose is oxidized to generate energy, amino acids are used to synthesize new proteins such as enzymes and hormones, and lipids form a major component of plasma membrane. As the food passes through the alimentary canal, it is broken down in stages, 3 stages, until the digestible material is dissolved and absorbed. Meanwhile, the indigestible residue is removed through the anus. Digestion involves both physical and chemical processes. Physical digestion involves the breaking up of large pieces of food into smaller pieces by mechanical means. It starts in the mouth with the slicing and chewing action of the teeth, like this. It continues to a smaller extent in the stomach by the churning action brought about by the contraction of the muscles of the stomach wall. Chemical digestion. Digestive enzymes break down complex food molecules into simple soluble molecules which enter the bloodstream to be transported to the whole body. This process normally involves hydrolysis reactions. Just now I told you that we have 3 stages of digestion. The first start in the mouth, next stomach, and lastly, small intestine. Let's go to the first stage, digestion in the mouth. The digestive process starts in the mouth. The chewing action breaks the food into smaller pieces and increases the surface area of the food for digestive enzymes to act on. The presence of food in the mouth triggers the secretion of saliva by 3 pairs of salivary glands. Saliva contains the enzyme salivary amylase, which begins the hydrolysis of starch to maltose. The thoroughly chewed food is rolled into a mass called a bolus in preparation for swallowing. This is the bolus. During swallowing, a cartilage flap called the epiglottis temporarily closes the airway to prevent food from entering the trachea. Next, The bolus enters the esophagus, which is a muscular tube lined with the epithelium and mucous glands. The mucus lubricates the bolus and aids the movement of the bolus along the esophagus by peristalsis, a series wave-like muscular contractions along the esophageal wall. That is peristalsis. When the cardiac sphincter relaxes, the bolus enters the stomach. Cardiac sphincter is a ring of muscles which control the opening of the stomach. Next is the second stage, digestion in the stomach. What is stomach? The stomach is a thick-walled, sausage-shaped organ that is situated below the diaphragm. It is muscular sac with a highly folded inner wall. The epithelial lining of the stomach contains gastric glands that secrete gastric juice. Gastric juice contains mucus, hydrochloric acid, enzymes pepsin, and enzyme renin. My formula is RAP. R stands for renin, A acid hydrochloric, or in BI hydrochloric acid, and P pepsin. Hydrochloric acid creates an acidic condition which the pH is pH 1.5 until 2.0, which is optimal for the action of the enzymes in the stomach. It also stops the activity of salivary amylase and helps to kill bacteria in the food. Pepsin starts the hydrolysis of large protein molecules into smaller chains of polypeptides by breaking specific peptide bonds. Renin coagulates milk by converting the soluble milk protein caseinogen into insoluble casein, and mucus, secreted by the goblet cells of the gastric gland, protects the stomach wall from the action of hydrochloric acid and digestive enzymes. Food stays in the stomach for a number of hours. During this period, the food is thoroughly churned and mixed with the gastric juice by the peristaltic contractions of the stomach wall. Eventually, the contents of the stomach become a semi-fluid called chyme. Relaxation of the pyloric sphincter allows the chyme to gradually enter the duodenum. Now we have the last stage, which is digestion in the small intestine. The small intestine consists of duodenum, jejunum, and highly called ileum. The duodenum, the first part of the small intestine, receives chyme from the stomach and secretions from the gallbladder. The liver secretes bile, an alkaline greenish-yellow liquid. Bile is stored in gallbladder. Bile does not contain any digestive enzymes. It creates an alkaline environment, which the pH is between 7.6 until 8.6 for the enzyme action in the duodenum. It helps to reduce the acidity of chyme and optimizes the pH for enzyme action. Bile salts emulsify lipids, transforming large lumps of lipids into tiny droplets, thus providing a greater surface area for digestion by enzymes. This allows a lipid digestion to proceed more rapidly. Then bile enters the duodenum via the bile duct. Pancreas. Pancreas secretes pancreatic juice, which contains the enzymes pancreatic amylase, trypsin, and lipase. The optimum pH required for the action of the enzymes in the pancreatic juice is between 7.1 and 8.2. Pancreatic juice is secreted into the duodenum by the pancreas via the pancreatic duct. in the duodenum, the digestion of starch, protein, and lipid takes place. The pancreatic amylase hydrolyzes starch to maltose. Trypsin digests polypeptides into shorter chains of peptides. And lipase breaks down lipids into fatty acids and glycerol, which are small enough to be absorbed by the epithelial lining of the small intestine. The formula is TALI. T stands for trypsin, A stands for amylase, and L stands for lipase. Next, it will pass through the jejunum and we enter the ileum. The glands in the wall of the ileum secrete intestinal juice, which contains digestive enzymes, are needed to complete the digestion of peptides and disaccharides. The formula is MEL, sucrase, maltase, iriopsin, and lactase. The intestinal enzymes require an alkaline medium to act at an optimum rate. At the end of the digestive process, all carbohydrates are digested into monosaccharides such as glucose, fructose, and galactose. Meanwhile, proteins are digested into amino acids and lipids into fatty acids and glycerol. Carbohydrate digestion. Maltose is hydrolyzed by maltase into glucose. The digestion of the other disaccharides, each of which is catalyzed by its own enzyme, is as follows. Sucrose is hydrolyzed by sucrase into glucose and fructose. Meanwhile, lactose is hydrolyzed by lactase into glucose and galactose. For protein digestion, peptides are digested by iriopsin or a peptidase into amino acids. Remember, vitamins and minerals are extremely small and soluble and need not be digested. Meanwhile, dietary fiber cannot be digested in the human body as the enzyme cellulase is not produced in the human alimentary canal. That's all from me. I'm sorry for any kekurangan. Thank you.